All right. So this is Dr. Fry, and this is my lecture on meiosis, which is Chapter 7, The Cellular Basis of Inheritance in the OpenStax book. So I'll just start out by showing you these three forms of reproduction. So the first one is binary fission. This evolved first. Most prokaryotes do this, or single-celled organisms. They just have one chromosome. All they have to do is duplicate that chromosome, and then the cells separate. So they're really good at reproduction. If you get a bacterial infection, it can get bad really fast. So they can do lots of cell division. Mitosis is what we covered last chapter. So this is where you're starting with a diploid cell and you're ending with two exact copies of the diploid cell. So these are considered two daughter cells, and they should be exactly the same as the mother cell you started out with. They go through S phase. That's where the DNA gets replicated. That's where you have the leading and lagging strand and the Okazaki fragments and all that. Here they're on the metaphase plate. There's anaphase. Then you should have two exact copies of this cell up here. What we're going to do next is meiosis. And meiosis basically goes through G1 and S, where it's replicating the DNA just like it did over here. And then it goes through G2, but then it's going to go through meiosis, and it's actually going to go through reduction because your gametes only are haploid, right? They only have half as many chromosomes as your somatic cells. Okay. Let me move this up here. All right, so what you should be able to do is define meiosis, describe the phases of meiosis M1 and meiosis 2, and describe the crossing over and summarize its purpose. So DNA replication occurs during this S phase right here, and this is the same as it was this is the cell cycle for mitosis. If it was meiosis, it would have more steps here. But the S phase is the same, right? So the G1 phase is the same, the S is the phase, same, and G2 is the same. The only thing that's different is instead of mitosis, we're going to do meiosis. But the cell spends approximately 90% of its time in interphase, where it grows. It has to replicate its organelles. It has to make more DNA. It has to make sure it has all the nutrients and everything it needs to make new nucleotides. So there's basically two types of cell division that can occur. The first one we did was mitosis. So this is asexual. So basically, after you are an egg, you get bigger because of mitosis, right? So you're just making more of yourself. And like grass growing is asexual. So grass is doing mitosis all the time. When you mow it off, it grows back. When you mow it off, it grows back, right? So that is asexual. Meiosis is just when you do sexual. So like flowers are sexual parts of the plant. They can grow asexually all the time. Only flowers are really the sexual part of the plant. And again, these are the stages of mitosis that we just went over. And then these are the phases of meiosis, which it's going from a 2N cell to 4 1N cells. So, and we'll show you those steps there. I go through them here. We'll talk about chromosomes, and we've done this a lot, but I'll do it again here. So DNA are thread-like structures, and it's made out of this double helix, right? So the backbone is the ribose and the sugar, and then these four are the nitrogenous bases. So C always goes with G, and T always goes with A. And then they get coiled up by these histones or proteins, and then they get coiled up more, and eventually they get coiled up into the chromosomes. But when they're in this form, you cannot make a protein out of them, right? When they're uncoiled in chromatin, you can make proteins, and you can get copied. It can be copied and things like that. So you really want to keep your your DNA and chromatin and not chromosomes, so you can actually have access to it. Okay, so each species has a fixed number of chromosomes. So all body cells are called somatic cells, and they all will have the same number of chromosomes. So in humans, this is 46. This means that a human cell in the brain, the finger, or the bone cell will all have 46 chromosomes. So every time you make a new cell, you have to make a full complement of your DNA, and you don't really want to make any mistakes on that. Okay, so the total number of chromosomes in a somatic cell is referred to as diploid, which means it has 2 copies of each chromosome. So we have 23 pairs. So 2 times 23 is, we have 23 different chromosomes and we have a pair of each. So 2 times 23 is 46. So chromosomes are arranged in pairs. So you get one from your mom and you get one from your dad. And they are should be the same length. They have the same position here where the centromere is. They have banding patterns. And they carry genes for the same traits, right? So if the gene for hair color was here, the gene for hair color would be here. If the gene for eye color was here, the gene for eye color would be there. So these are homologous chromosomes. They have the same chromosomes in the same place, and they're all the same. So they're the same. Homo means the same. Okay, so humans have 46 chromosomes. They're arranged in 23 pairs. So they're called a homologous pair, and you inherit one from each of your parent. And this is a karyotype. So these are your sex chromosomes right here, right? So these can be different. If you're a boy, you're different. So you have an X and a Y if you're a boy. But if you're female, you'd have 2 X's and they would be homologous. Okay, gametes. Collectively, egg, sperm, and pollen are referred to as gametes. Gametes are specialized haploid cells, so they have 1 copy of the chromosomes. Okay, so that means they have half the number, so they're haploid. Okay, in humans, the number of chromosomes found in a gamete is 23. So that when the sperm fertilizes the egg, the total number of chromosomes in the zygote will be 46. So if you had a 2N gamete and a 2N gamete and they went together, then your offspring would have 4N. And so you have to do the reduction in order to get back to the original 46 chromosomes. So that's why you have to do the chromosome reduction. All right, so, and you have to go through meiosis too. So, okay. So a change in the number of chromosomes in a cell can lead to a mutation in the cell. We already covered that stuff. All right. So we'll answer these. So draw and label a pair of homologous chromosomes. So they're over here on the side, right? So here's the centromere for these. You get one from your mom and one from your dad. They have the same exact genes on the same, these are called loci, is sort of where it is. Give an example of a haploid and diploid cell. So a haploid cell for us will be the egg or the sperm, and the diploid would be, your hair cells, your skin cells, your fingernail cells, your eye cells. State where half of your chromosomes come from. So you get half from your mom, and then you get half from your dad. And state how many chromosomes are in a lung cell. That would be 46. A sperm cell would be 23. And an egg cell, well, this is an ovary. An egg cell would be 23 as well. Okay. Okay, so meiosis is a cell division, which forms gametes. And so you're going to move from 2N to 1N. So it's a reduction of chromosomes. So like mitosis, meiosis begins with meiosis begins with DNA replication, which happens right here at this S phase. Um, but they want to have the chromosome, so you're going to have to go through 2 rounds of these stages here, right? So you're going to have meiosis 1, and then you're going to have meiosis 2. They're going to go through those same stages twice. And I'll just remind you, this was the replication, right, during the S phase, where you have the leading strand and things can be added to the 5' end, but here they have to add them to this end, so they're actually being backstitched. They're being backstitched this way. All right, so that was that. So the overall process of meiosis is shown below. So interphase includes that G1, S, and G2. And here is where you're going to have these tetrads basically going to come together, and then you're going to have 2 daughter nuclei. Then you're going to go through meiosis 2, and you're going to get 4 haploid gametes right here. So this has half as many as this, right? So this cell has half as many as this cell. And you can see also that they are different, right? So so here it's showing you get the white ones from your mom, and the black and the red ones from your dad, and then you have crossing over here where this is where you get a mix up, right? So there was a crossover between these two, and there was a crossover between these two, and so that gives you different genetic variation, and that's why you look like your parents and you look like your siblings, but you don't look exactly like your parents and exactly like your siblings. So you have the same genes, they're just you know, the way they cross over makes you look different. All right. And then you have 4 gametes that actually look different than the parent cell as well. Okay, so they're going to go through this step by step. So in meiosis, the separation of the homologous chromosomes and sister chromatids are accomplished in 2 stages. So this is meiosis 1. So in meiosis 1, the cell undergoes many processes to prepare itself for, so this is where this happens during this S phase right here. So basically what's going to happen is each chromosome is going to get copied, right? So this is 2 copies of this chromosome. These are called sister chromatids. Here's a copy of this one copied, and these are called sister chromatids. But these are actually going to hook up, right? So these centromeres are actually going to hook up, and they're actually going to be called tetrads. So tetrads mean 4, right? So these 2 are actually going to go together, which did not happen in mitosis. Okay, so basically those homologous chromosomes, that tetrad is going to get separated in 4 phases. In prophase 1, the DNA condenses into chromosomes and they line up together in pairs of tetrads, right? So this is that tetrad right here. The homologous chromosome, they cross over and exchange genetic material in crossing over. And right here, it's going to cross over here, right? So now the red went on the blue and the blue went on the red. And there's all kinds of crossing over that can happen. It can happen lots of times. It can happen not lots of times. You know, it's random, basically. And so, but this is a recombinant chromosome then, right? So this happens when they're right here. When they're hooked together, that's when this crossing over happens. And so this allows for variations in traits in the offspring. The spindle fibers are going to attach to the chromosomes, right? So you still have the spindle fibers coming from the centromeres or the centrioles, and the spindle fibers are going to attach to the centromeres, and the nuclear membrane right there is going to disappear. So the tetrads arrange themselves along the cell's metaphase plate, and that's right here, right? So they're on the metaphase plate, which each member of the homologous pair oriented towards the opposite pole. And then, so here this green is facing this way and this red is facing this way, but they actually could be switched, right? And the same thing here. This one could be attached this way. The red one is attached this way and the green one this way, but they could be switched, right? So the green could attach that way and the red could attach this way. So that's independent assortment, and I'll show you that in a minute. Okay, so yeah, the chromosomes are arranged randomly. So each gamete will contain a mixture of maternal and paternal alleles. So if all the maternal ones were, say, they're green, they lined up on this side, and all the paternal ones were red and they lined up on that side, that would be, you'd have things that look just like the mom and the dad. But actually they line up independently. So they can switch either way, right? So they can line up either way. And that's called independent assortment. So here's another example, right? So we have 23 pair, but here's just showing you 2. So here is where all the moms are lined up here and all the dads are, this is maternal. So all the moms are lined up here and all the dads are lined up here. So they are going to form haploid gametes that look like that, right? So basically this one's going to look just like the mom and this one's going to look just like the dad. But here they lined up differently, right? So this one actually lined up the same, but this one here lined up differently, right? So now the red is on this side and the green is on that side. So then when they get pulled apart, this is going to be from the mom and this is going to be from the dad. This is going to be from the dad. This is going to be from the mom. And then when you separate them again, you are going to have those gametes, right? So now these gametes look different than these gametes. So you can imagine, that gets decided 23 times. So there's 23 pairs. So we do have 23 pairs of chromosomes. So that would be, they can be on one side or the other. So they can have 2 sides they can be on. That's where the 2 comes from. And then you have 23 pairs. So 2 to the 23 pairs is actually more than 8 million possible combinations of chromosomes in human gametes. So there's over 8 million different possible ways they can line up on that plate. Okay, and that's where they say no 2 humans are identical. So the idea of two meiotic events having the same crossover and the same independent assortment is highly unlikely. Highly unlikely. Okay, so then anaphase 1 is when the spindle fibers shorten and the homologous chromosomes are pulled to the opposite sides of the cell and separated. But the sister chromatids do remain attached. So the cell membrane begins to form along the center of the cell, right? So that's right here. And right, so this one came from the mom and this one came from the dad, right? So this one's going to have different looking gametes. And then this is when they separate. So right, so at the end of meiosis 1, the spindle fiber disappears and a new nuclear envelope forms. Cytokinesis takes place during telophase 1, which results in 2 unique cells, right? So this one is, they do look different than the mother cell because you got one from your mom and you got one from your dad on each of these cells. All right. So the cell now undergoes a resting time in which they unravel their chromosome. So it goes back into chromatin. They then enter meiosis 2 without undergoing DNA replication. So they're not going to do any more replication. So during meiosis 2, the sister chromatids are separating, generating 4 unique haploid gametes. So this is going to show you meiosis 2. So this is during interphase after meiosis 1, right? So these are these 2 cells. You got 1 chromosome from your mom and 1 from your dad for each one of these. Okay. Prophase 2, the nuclear envelope disappears and the chromosomes condense. The spindle fibers appear and attach to the chromosomes, right? So here's the spindle fibers being formed here. And like mitosis, the 2 sister chromatodes are attached to the spindle fibers from opposite poles, right? So they're going to pull them apart at those poles eventually. Metaphase 2 is the chromosomes align individually on the equator, so they're not in tetrads anymore, right? So last time they lined up, they were in tetrads. Now they're just sister chromatids. So this is so seen in so the way they line up here is also random. Okay, and then the spindle fibers contract, and the sister chromatids are drawn towards opposite poles of the cell, and then the cell constricts and between them. Okay, and then telophase 2 in cytokinesis, the cell membrane forms and the new cells pinch off, right? So this is the phospholipid bilayer around here, right? And this results in 4 new haploid cells, right? So you're going to have 4 haploid cells. They have half the number of, these are haploids, so these are gametes. Okay, so state the purpose of meiosis and give a brief overview of the process of meiosis. And the purpose of meiosis is to produce gametes. All right, so that's it, to produce egg and sperm. And then there's the answer to that. And I think that is it. So that wraps up that. I hope that's helpful.